Let's jump right into the explainer. When we look at a forest, or honestly, even just a potted pothos on the windowsill, it looks incredibly still, right? Almost totally static. But beneath that calm, green exterior is a bustling, high-speed highway of water, minerals, and nutrients. Today, we're unpacking exactly how that hidden mechanics works. To make sense of all this dense botanical physics, we've got a pretty structured roadmap for this explainer. We're going to trace the history of plant physiology, outline the grand mystery of plant transport, open up the cellular toolkit, and then dive deep into the invisible forces running the show, specifically water potential, osmosis, and imbibition. Let's kick things off with the major shift in how we actually study plant physiology. For a long time, biology was all about ecology, looking at the organism as a whole. But eventually, a completely new perspective emerged that zoomed way in on the cellular and molecular level. A perfect example of this is Melvin Calvin. Picture this, it's just after World War II. The world is still reeling from the atomic bombs. But Calvin and his team decided to take radioactivity and use it for something incredibly positive. He used radioactive carbon-14 to literally map out how green plants form sugars from carbon dioxide, water, and minerals. It was a massive breakthrough, earning him a Nobel Prize in 1961, and it proved that to really understand a plant, you have to look at its invisible, internal chemical pathways. And that molecular perspective brings us face to face with a massive biological puzzle. It's a logistical nightmare that scientists grappled with for decades. Think about it. How do plants manage massive vertical water transport from deep underground roots all the way to the highest leaves? Animals, like us, have a circulatory system. We have a heart constantly pumping fluid around. Plants, they don't have a heart, no pump at all. Yet they manage to move molecules over distances far greater than most animals ever do, completely unidirectionally, straight up. How is that even possible? To solve this, we need to systematically unpack the plant's microscopic toolkit. We have to look at the exact mechanisms cells use to move stuff around on a micro level. First up is simple diffusion. This is a completely passive process. Imagine molecules just moving in a random, chaotic fashion, but slowly drifting from an area where they are super crowded to an area where they have more space. It doesn't cost the plant a single drop of ATP energy. It's slow, sure, but absolutely crucial because it's the only way gases can move within the plant body. But there's a catch. A plant's cell membranes are mostly made of lipids. So while fat-soluble stuff slips right through, water-loving hydrophilic substances just bounce right off. They need a VIP entrance. Enter facilitated diffusion. The plant uses specialized membrane proteins to act as open doors. For instance, aquaporins are these incredible proteins that form water channels. The protein binds a molecule, physically rotates, and drops it inside the cell. Again, no energy cost here, just riding that natural concentration gradient. What's fascinating is how these carrier proteins actually manage the microscopic traffic. Some of them act like a buddy system. They only allow diffusion if two specific types of molecules cross together. If they both go the same direction, we call it a symport. If they trade places, moving in opposite directions across the membrane, that's an antiport. And if a molecule just rides solo, that's a uniport. But what happens when the plant needs to push molecules the wrong way, from a low concentration up into a highly concentrated area? It can't just rely on natural passive diffusion anymore, it has to force it. This is active transport. The cell literally uses ATP energy to power specialized pump proteins to carry substances uphill. And just like enzymes, these pumps are super specific and they max out once all the transporters are fully saturated. So if we look at it all together, the differences are pretty stark. Simple diffusion is just a passive free-for-all. Facilitated transport brings in those selective special proteins, but it's still a free ride down the gradient. It's only active transport that can do the heavy lifting, pushing substances uphill but demanding precious ATP energy to do it. Okay, so we've got the transport methods down, but what's the actual engine driving massive volumes of water through these pathways? To get plant-water relations, you really just need this one secret code. Water potential equals solute potential plus pressure potential. Here's the deal. Water molecules have kinetic energy. They are constantly buzzing around. Pure water has the absolute highest water potential, which, just by convention, scientists set to 0. If you dissolve something into that water, you reduce its free energy, dragging the water potential down into the negatives.
But if you apply pressure, the potential spikes up. The golden rule you need to remember is that water will always flow from an area of higher energy to an area of lower energy. And when you combine that water potential with those selectively permeable membranes we just talked about, things get really powerful. We get osmosis. This is the spontaneous diffusion of water across a membrane, completely driven by those pressure and concentration gradients. And depending on the environment outside the cell, osmosis physically changes the plant. If the outside balances the inside, it's isotonic. The cell's just kind of flaccid. But if you drop that cell into a hypotonic, dilute solution, water rushes in. The inside swells up against the rigid cell wall, building what's called turgor pressure. This is a huge deal. It's literally what forces plants to grow and extend. On the flip side, put it in a highly concentrated hypertonic solution and water rushes out. The inside shrinks away from the walls in a state called plasmolysis. You can actually see this osmotic pressure in action with a really cool kitchen experiment. If you dissolve an eggshell in dilute hydrochloric acid, the hard shell vanishes, but that weird, thin, semi-permeable membrane stays perfectly intact. Tie that membrane to a funnel filled with a sugary sucrose solution, flip it upside down into a beaker of pure water, and just watch. Water from the beaker, which has the higher water potential, will push its way across the egg membrane into the sugar water. You will literally watch the fluid level in the funnel creep upward, totally defying gravity, until it hits equilibrium. It's wild to see water potential in real life like that. Now let's zoom back out to the macro scale and see how these tiny forces shape the actual world. We just saw water cross a membrane, but there's another type of diffusion that is borderline explosive. It's called imbibition. This happens when solids or colloids absorb water and just massively swell up in volume. Think about a dry, tiny seed in the dirt. When it absorbs water, the sheer pressure of imbibition is what gives that little seedling the brute strength to break through heavy, packed earth. It's so powerful that prehistoric humans actually weaponized it. They would jam completely dry wood into the cracks of giant boulders, pour water on it, and wait. The swelling pressure from the wood imbibing that water was literally strong enough to shatter solid stone. Which leaves us with a pretty wild thought as we wrap up this explainer. We look at a tree and just see a piece of scenery. But inside, driven purely by the invisible math of water potential, the careful gating of aquaporins, and the literal rock-shattering pressure of imbibition, water is constantly surging upward from the dark soil into the sky. If these invisible cellular forces can break boulders and pull water hundreds of feet in the air without a pump, it really makes you wonder, what other immense, silent powers are operating right beneath our feet?